Hi everybody, we're going to talk about how do you prevent Alzheimer's, and really what we're talking about is how do you prevent dementia? Why are so many old people cognitively impaired, and what could you do to reduce your risk of ending up like that? Okay, help your parents and stuff. One of the things I do is I'm a neuroradiologist. I look at a lot of brains. It's very common for me to get a request. Memory problems, cognitive impairment, probable Alzheimer's, please exclude other causes. Okay, and I look at these brain MRIs and I'll tell you what I see. I often get to compare their old brain MRIs going back 20 years or more. And I'll say, gee, this person's brain keeps shrinking. Now classically people think of people becoming demented from Alzheimer's, for example, or they say the person had a big stroke and they were kind of demented after that. I can tell you that's not the most common cause. The most common cause is brains just shrink. Why do people's brains shrink? They look pickled sometimes. Alcohol will do that. Alcoholics just pickle the brain. It's totally stupid to drink alcohol. Don't drink any alcohol. Not one drop is what I recommend. But anyways, besides the direct toxic effects of alcohol, which is, there's not that many alcoholics, brains shrink. And when you see a stroke is a blockage of an artery with immediate loss of blood supply and oxygen to the brain tissue, so it can't make any ATP. It's a sudden death of a brain cell. And when that occurs, the plasma membrane lyses. All the contents of the cell are spilled out into the extracellular matrix, and that activates a big microglia, the brain immune system cells response. There's all this edema, meaning leakage of fluid, and you see a big stroke on a brain MRI. I can see that in 2 seconds. Brain stroke right there, and tell you exactly where it is. That's easy to see. But that's not what happens most of the time. I just see a shrunken brain. Sometimes I don't even see any strokes. So what is happening? There's different types of cell death. So a stroke is called necrosis, with lysis of the plasma membrane. But a more common form of cell death is what is called apoptosis. Apoptosis is programmed cell death, whereby the cell in a sense recycles itself. So sudden complete loss of blood supply and oxygen, cell dies. Necrosis, stroke. But more often it's a partial decrease in oxygen, a partial decrease in glucose supply. The cell can no longer make an adequate amount of ATP, but it is able to send a signal through its mitochondria to recycle itself. And then the plasma membrane does not lyse, so there's no edema. You don't see any evidence on a brain MRI or even at autopsy of what happened to that neuron. It's just recycled and gone, all right? So you don't want your brain cells going into apoptosis. Why do brains shrink? The main reason is apoptosis. And I said, well, why does apoptosis happen? And this guy, Peter Quo, wrote some great papers back in the 1950s and 60s. And they did some interesting things. Back in those days, before the IRB, you could do whatever you wanted for experiments almost. Took a bunch of cardiac patients with cardiac angina, chest pain due to ischemia, blockage of arteries. Fed them a high-fat meal. Checked their blood lipids every half hour, and what they found is peak lipemia, maximum levels of fat in the blood, was around 5 hours. And guess what? That's when they got chest pain, okay? They're plugging up their arteries. A guy by the name of Rosenman did a beautiful paper in 1964, I think it was a journal of circulation, and they put an 80 times magnification microscope over a person's eye, and they could see the little tiny capillaries and arterioles occluding in real time, all right? Now you got to figure most of those probably opened up because the person doesn't suddenly go blind typically. But the point is, when you eat a high-fat meal, especially if you superimpose sodium, salt, it'll vasoconstrict and clamp those arteries off. You're going to be losing some blood cells. And the eye is not just a window to the soul, it's a window to the brain. The same thing's happening in the brain, okay? And you're plugging up those capillaries. I've seen that in different types of mouse studies as well. And now that brings up a very interesting guy here, Dietary, all right? This guy, Dietary, is a PhD scientist. He wrote this book, Alzheimer's Turning Point. And what he would do is he would take mice. I know it's a crappy drawing of a mouse, I drew it, but he would tie off the carotid artery. See it being tied off? Tie off the common carotid artery. Here's, let's say, is the external carotid, internal carotid, goes up to the brain. And the mouse would then become demented in about 2 months. Then they would sacrifice the mouse at autopsy. They look at its brain, and usually, guess what? No stroke, okay? But the brain is shrunken. Apoptosis, okay? So by having a lack of blood supply and glucose supply to those neurons, they're gradually dying, but slowly enough that they can recycle themselves, so the brain shrinks and the mouse becomes demented. He experimented with tying off the internal carotid artery, the vertebral arteries, etc. And so then you say to yourself, well, gee, is that perhaps a good metaphor or way of viewing cognitive impairment in general? What else happens? We've all seen patients with atrial fibrillation, abnormal heart rhythm, all right? They're not effectively pumping blood. We typically think of AFib as causing cognitive impairment by blood clots will form in the heart. They get tossed up to the brain. It's called an embolism. Person has a stroke and they're cognitively impaired. But guess what? That's not the main reason AFib causes cognitive impairment. It causes cognitive impairment because he's like the mouse. They're not getting enough blood supply to the brain because they have abnormal cardiac pumping, okay? Guess what? Hypertension, super common. A typical hypertensive person goes, oh, it's no big deal, it's under control. Yeah, right. Hypertension is very dangerous. If your pressure is too high, you get little bleeds in the brain, cerebral microbleeds. You also get what are called the hypertensive lacunar infarctions. You can also get a big bleed, intracranial hypertension, intraparenchymal hypertension. Hypertension is dangerous, okay? And you got a narrow therapeutic window you got to stay in to keep the brain adequately perfused. If you overtreat your hypertension, guess what? You're like the mouse. You don't got enough blood supply to your brain. You're chronically underperfused and you're losing a lot of brain cells, all right? So my advice, if you're hypertension, you're probably fat, you're probably eating a lot of meat, a lot of salt, and a lot of fructose. Stop eating that. Be careful. Titrate your meds down. Let your doctor know. All right, what else? What happens with the high-fat meal? High-fat meal, saturated fat in particular, meat in particular, but also some of these oils. They'll increase LDL cholesterol. Fructose will also do this. Alcohol will also do this. LDL cholesterol is a bridging molecule. It sticks the red blood cells together. Red blood cells on their outer surface have sialic acid residues that have a negative charge on them. And that's part of what is called their glycocalyx, all right? It's like trees sticking up on the surface of a mountain. They have negative charges on them that repel other red blood cells. But bridging molecules like LDL cholesterol, fibrinogen, a blood clotting protein, IgM antibody, especially in acute infections, and uric acid is also thought to be a bridging molecule. They will stick the red blood cells together. Red blood cells stuck together are a rouleau formation. That means stack of coins in French. Typical red blood cell is about the same size as a capillary, 5 to 7 microns. So when they're all sticking together, they become big and bulky, hard to get through those capillaries. And that causes increased blood pressure because your heart's pumping a milkshake, in a sense, thick blood instead of pumping thin blood, more like water. All right, so you don't want to eat that. You don't want to be eating meat. Don't eat any meat, don't eat any oils, and avoid any sweetened things with fructose. Alcohol in large amounts also has a similar effect, causing increased saturated fat, because a 2-carbon unit of ethanol can get made into acetyl-CoAs, which is how you make fats. All right, what else causes dementia? Brain damage, makes people stupid, demented. It's sleep apnea. Tons of fat people have obstructive sleep apnea. They get thick, soft, fleshy tissues in the back of their throat, and their airway can partially plug up while they're sleeping. Not good, okay? Because guess what? They're hypoxic. Nowadays, they can take these sleep apnea patients and put them in a sleep study and measure their pulse ox, oxygen saturation, all night long while they're sleeping. Guess what? A lot of them are dropping their O2 saturations. Normal is, by the way, 95 and higher. 95 to 100 is O2 sat percentage, all right? When they start going into the 80s prolonged amounts of time, they're losing brain cells, all right? And they often do that every day. So a lot of sleep apnea patients, you talk to them and they're really mentally slow. What else? When people are fat, they get something called Pickwick syndrome. It's from the Charles Dickens novel, Pickwick Papers, written in like the 1830s or something. And basically a fat person, the fat in the belly, it's a compartment. And the adjacent compartment is the thorax. And the lower the diaphragm, big fat belly impedes lowering of the diaphragm a little bit. So just in general, like you've seen a lot of fat people, they get kind of short of breath just going up a couple stairs. Okay, diabetics. Diabetics nowadays can get CGMs. That's a continuous glucose monitor. And this is unique because in the past, a diabetic would just do a finger stick a couple times a day to get a sense of where their glucose is at, determine their insulin dose, for example. But what I'm saying is nowadays they can get a continuous glucose monitor and get a printout of their blood glucose level every 30 minutes through the night. Guess what? A lot of them are hypoglycemic, have low blood sugars at night. They're like the mouse, okay? They're not getting enough glucose to their brain cells for a prolonged amount of time. They're losing brain cells, and they better figure out their glucose, otherwise they're going to end up demented. All right, so what would you do? Like if I was diabetic, what would I do? Number one, I would figure out with my CGM exactly where my sugars were. Number two, I would not eat one bite of meat, not one bite. Never drink fructose. Never, ever drink that stuff because you don't want saturated fat. Saturated fat causes insulin resistance. It impairs mitochondrial function and skeletal muscle, leading to insulin resistance. The GLUT4 transporters can't go up to the membrane, so you get hyperglycemic, and you start this whole cascade of negative events. We'll talk about that in my diabetes lecture, but if you avoid that, then your carbohydrate tolerance goes way up. You eat a plant-based diet with starch, a lot of fiber, you'll get the best possible results with a diabetic. All right, hypertension, we talked about it, AFib, we talked about it. So anyways, don't end up like the mouse, all right? And the way you do that is eat a starch-based, plant-based diet with no oils. Don't be drinking alcohol or sweets. Get your sleep, and you'll have the best chance of lowering your risk for ending up demented at an early age.